Hi everyone, welcome at the Innovation Café. My name is Margot, I'll be hosting, and welcome back to the people that are here live again that we saw before the lockdown and everyone that's new. We're very happy to be live again, but we also have people joining us online, so I also want to welcome the people that are watching. For the people that haven't been here before, we're the Innovation Café, we're a networking event for the tech design and knowledge community here in Eindhoven. And basically what we do is we want to show people what happens here in these fields, because a lot of the stuff is behind closed doors, and we want to open this up for the audience to discuss and to see what innovations are happening in the region. We did this together with a lot of partners. Today, no partners involved, but I just want to mention them that we couldn't do that without them. And afterwards, we always have a networking drinks as well. So also for the people that are joining us online, we hope to see you at some point live again. We can imagine that you might be waiting for vaccinations, or you want to wait it out a little bit, but we hope to see you at some point here in Kazerne. So I just want to point out that we're here every week. So we have a weekly program, and so you can look it up on the website, ehvinnovationcafé.org, to see all the future program for the rest of the year as well. And for now, I want to point out that we have a speaker here from iGem, Luc Rossen, and he's going to talk a little bit about their project. So for now, I want to give the word to Luc, and I'll see you after.
Yes, thank you, Margot. As you said, my name is Luc Rossen. I'm the team manager of Team iGem. We're a student team at the TUE. Let me get my clicker. All right, test. So, I kind of want to start with explaining iGem, because a lot of people might not know about this. It's a worldwide competition in which we participate. Over 50 countries in the world participated, over 6 continents. For example, this year around 5,000 teams are participating. It's on one hand a very networking type of event. We collaborate with teams all over the world, but we also compete for medals, for example. And what we try to do is we are free to identify a problem in the world, usually biomedically oriented, and we try to find a solution for it in the context of genetic engineering. So that's what iGem also stands for. It's the international competition for the genetically engineered machine. So that's what we try to solve, or that's what we try to participate on. As I said, over 5,000 attendees this year we expect to be joining, and it's going to happen in Paris this year if Corona allows it for it to happen, which will culminate into a giant jamboree, which is like a giant competition where all the teams come to present their work, to show what they've done, and to see who earns what reward. So our team, a little bit about us. I'm the team manager. My name is Luc Rossen. Our team is a little smaller than the conventional teams. We are 8 in total. We have some guests here as well from our team today. What we want to make up for in quantity, we want to make up for in quality. We have 8 team members. Each of us is currently a student at the TUE, a first-year master student. For example, I myself, I'm a student of biomedical engineering, chemical biology, under Luc Brunsveld. So, in essence, this is supposed to be a competition for, how would you say this? Hello? 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 Okay. So, the idea is that the teams are supposed to be interdisciplinary. However, we are strongly a team from the biomedical engineering side. However, I would like to point out that because we want to be as autonomous as possible, we serve as what we try to do is we try to solve this as a biomedical engineering problem, but we also have to be autonomous in gathering sponsors, in setting up our infrastructure. We need to plan things, we need to organize events, we need to work on human practices, for example. So beyond just working on the project, we also try to diversify ourselves, develop ourselves in roles that you often see in businesses, for example. So that's also something this competition tends to offer. But beyond our team, we also have some support from the university itself. For example, on the left you see our advisors from the faculty. For example, Luc Brunsveld, Maarten Merks, Tom de Geef. These are professors in particularly biomedical engineering as well. And what they do for us is essentially when We try to be as autonomous as possible in our work. However, because we work with, for example, genetically engineered species, this is still very dangerous, or there's risks involved. So strong regulations exist, especially in Europe, for example. So these professors, they come in when we essentially need the regulatory part. You know, when we need to order, for example, last week, what is it, antibacterial spectinomycin. You know, we have to involve the professors, for example, for things like this. So what we want to try to say with this is that we try to be as autonomous as possible as a team of students, but we also employ the specialists when necessary. And for example, Claire, you see in the top, is an old team member, so we try to initiate the continuity, because each year a new competition starts, but we try to support each other as the old team. So for example, Claire is a team member from 2019. We are in direct contact with her to kind of pass on the stick from one year to the next, and kind of connect with all team members. So we essentially network outside of the university with other teams, but also internally in the university with all team members and faculty members. And for example, we also get support in the lab from, for example, Dr. Yanni, so that when new protocols need to be checked, for example, we have specialties or specialists for this as well. So now actually what are we doing? What is our case in this case? We identified inflammatory bowel disease as the problem we want to solve. In short, what it is just chronic inflammation of the intestine. Under this, maybe you've heard of it, like Crohn's diseases, for example, under IBD. Ulcerative colitis is one of them. So, for example, Crohn's disease is inflammatory, the inflammation of your entire gastrointestinal tract. Colitis, for example, is usually the large intestine. In this specific case, for example, the end of it. So the idea is these people, they suffer from inflammation in the intestine. They have to, they suffer from, for example, diarrhea. They have trouble eating. They have cramps. It's a very problematic disease, and perhaps more importantly, it's a disease that is modern. For example, 2 million people in Europe suffer from it currently, from which 90,000 exist in the Netherlands. This is roughly 1 in 200 people, so it's relatively serious. And also, it grows. It's not a disease that disappears as a society evolves. Some diseases can be regulated by, for example, hygiene, but this is actually one that's expanding. So look at diabetes, for example. This is also a disease that is prominent, thus warranting us to find a way to improve the life of the people who suffer from it. And then particularly, instead of the disease itself, we want to work on the diagnosis of it, because currently the diagnosis is highly invasive. For example, often it involves endoscopy, and then at the same time taking a biopsy. So what this essentially is you know, the physician, for example, has to insert the camera into a person's body, usually through an orifice, taking a biopt, which is highly problematic, or it's not a pleasant experience, I should say. And then particularly what is the case is that these people have to take laxatives for 2 days in advance, which is really unpleasant. They have to fasten, so they can't eat, they can't drink. For example, 8 hours before taking the endoscopy procedure, they can't eat anything, can't drink anything. So it's really, that's where we see the problem, because these people suffer from it. They have to have diagnosis, they have to have measurements taken, and we want to improve on that. So what we want to do is we want to make it non-invasive. So our project name right now in the top is IBD Tech this year. And how we envision it, finally, as a product, is the patient will ingest a simple pill, like a little pill you can take in your mouth, swallow it, it will pass through your stomach into your intestine, and there it will deliver live E. coli bacteria, or live bacteria. And in short, what they will do is they will detect a marker for inflammation, specifically for IBD, and then some translation cascade happens, and as a result, these bacteria, if they detect this inflammation, will produce gas vesicles. And then what you can do with the gas vesicles is the physician can, outside of the body, use conventional ultrasound equipment, so a simple transducer you see with pregnancy exams as well, monitor the stomach. You will have an image of the gas vesicles. Then you can excite them with the same machine to pop them, essentially, and then you can measure again. So what you have is then two images, one before and after popping, and that contrast is essentially analogous to how many gas vesicles were produced, which in turn we can deduce how much inflammation marker was detected. So what this essentially means, or what we expect for this to be, is that the physician can then, simply by ingesting a pill in a patient, detect the inflammation in the intestine, so something was happening deep inside your body, completely outside of your body, eliminating the need for taking a biopsy, eliminating the need for an endoscopy. We expect this to be relatively fast because you can just measure it directly and do the image processing immediately after, so there's no biopsy sample that has to be sent to the lab, which can take, for example, 2 weeks. And the physician doesn't have to do any particular procedure, so you don't have to, insert the camera in the patient, so you don't need specific training for that. And as well, the patient no longer needs to fast for 2 days and doesn't have to take laxatives either. So it's much easier to plan, it's much easier to perform. For the patient, it's much more, well, it's essentially non-invasive. That's what we envision. But what we do particularly, so not exactly the pill, what we do is what we want to proof of concept this mechanism in the cell, which you see in slightly more detail here. So what you see here is essentially the membrane of the cell. On the top you see the extracellular region, so that's essentially your intestine, and then the bottom part is in the bacteria itself. So what we want to do, we take this E. coli bacteria in this case, and we want to introduce 3 plasmids. The first plasmid translates for the top protein, the PTRS. So this is a receptor protein that will detect specifically this biomarker, and also in literature the specificity has been tested among other metabolites in the same order of molecules. So we expect this to selectively detect tetrathionate, which will induce autophosphorylation in the receptor. Then in the second plasmid we want to introduce is the reporter protein, the TTRR, the one in the middle. What this will do is it will bind to the phosphorylated receptor. It will in turn phosphorylate itself and dimerize with a copy of itself. And then this dimer will act as a promoter for our third plasmid, which you see in the bottom, which are the acoustic reporter genes. And what these genes essentially do when activated, or when produced, these are around 11 proteins, is they will oligomerize into hollow spheres that will permeate gas in the bacteria. So what you end up with is the detection of tetrathionate will induce the cascade that will result in a translation of proteins that will make these hollow vesicles. They will permeate gas, allowing for these structures to form. And then again, outside of the body, we can measure these because they have like a different refractory index, for example. You can then excite them at a specific frequency, which is unique to these specific acoustic reporter genes. And then you can measure again, and then you see exactly what was in the bacteria. And then the question is, for example, how many gas vesicles are produced for how much tetrathionate you detect. So we know, for example, from literature that that the tetrathionate receptor can measure it at a concentration of 50 micromolar, so it's selective enough or sensitive enough to detect the concentrations in the intestine. But what we want to do also is we want to model our project. For example, we use MATLAB for this. What you see here specifically is just our portion of the model that translates from the detection of tetrathionate to production of GFP, which is a fluorescent protein instead of the reporter genes. And what we want to essentially do with this is we want to make a prediction for, say you measure X quantity of tetrathionate, how much GFP is produced, and eventually we want to also make a prediction for how much gas vesicle is produced. And what we can do is we can couple this back to the lab to give them feedback on what concentrations do you work with your experiments, and finally also if you have your product, so say you measure Y concentration of gas vesicles, how much tetrathionate was actually in your sample. So that's why we use modeling kind of parallel to the lab to have an idea of the quantitativeness of our proof of concept. And I want to stress again, this is what we are directly working on. We want to in vitro test these bacteria, and then, for example, the translation to a product, so the pill, changing cell type, for example, is what we plan or perhaps could do in a PhD, for example. So currently we are concretely working on the proof of concept of this structure. But that was really our project. What we also do in the competition, as I mentioned before, is we collaborate with different teams. For example, what we now already have done is we collaborate with Aachen a city in Germany, or a team from Germany in the city Aachen. Our collaborations can range from simple online meetups, of course with Corona right now, to more thorough collaborations with Vienna, for example. The way we identify these things is we communicate with other teams through platforms organized by the competition. Then we can kind of exchange. For example, with Aachen, it was because we are locationally close to each other, so we thought it would be interesting to see if there's collaboration opportunities. But for example, with Vienna, what we want to do is their problem, what they had identified this year, is not the diagnosis of IBD, but the treatment. So essentially what we want to do with this team is we want to connect and see how can we provide, because our diagnosis is what is used for the treatment of IBD eventually, is how can we shape our project in such a way that you can use it as a diagnosis and vice versa. And what we also want to do is, because we collaborate and not just compete with other teams, is we want to do outreach to patients, for example, or to doctors. And our reach is, of course, limited to the Netherlands primarily, but Vienna can reach in Austria. So what we want to do is also exchange information and contacts in that way, where we essentially increase the range of feedback we can get for our project and shape our product eventually to a greater audience of patients and doctors who eventually want to use it, increasing our potential, hopefully. But also human practice is a big part of the competition. Human practices, you can roughly subdivide that into, for example, integration, so approaching patients and seeing how we can improve our own project for this, but also education and communication. And because we are all from biomedical engineering, we thought it would be interesting to see, can we appeal to the younger audience perhaps, so elementary school, because I personally, for example, in elementary school, in high school, I always felt that biology and chemistry specifically were always a bit lacking. They're usually not taught in the detail they exist in, and there's just so much more to it than that is usually taught. So we thought it would be interesting, also because team members of us, they work with elementary schools, to give guest lessons. And specifically, also in the context of the pandemic, it's not in the image here, but one of the guest lessons we did was showing why it is important to wash your hands with soap. So for children, it's an everyday appliance that you use, but understanding it can very often be the decisive factor in doing it or not. And it can also help to educate people on why actually do we use soap, or how does it work. So we thought, it would be fun to motivate children, see if they are, more appealing to biology, if they understand it better, and at the same time help people, fight the pandemic. And also what you see here, for example, with the child's face is, they enjoy it. So hopefully we spark some motivation into getting children to go into biology like us. And now, actually, we are just on the verge of going into the lab. We recently ordered the plasmids. We are still waiting for the delivery, but the idea is hopefully in the near future we can actually start. What we want to do is we want to set up our experiments in such a way that we first validate the plasmids, of course. We want to test the receptor cascade. We want to test the reporter genes, so the gas vesicles separately, and then finally integrate them. And then hopefully, parallel to that, we do modeling to see how are we expecting to work with what concentrations. And at the same time, we work on gathering sponsors, human practices again. So it's a lot going all together. And hopefully now we can go into the lab in the coming weeks. So I just want to do a quick thank you to the sponsors currently as well. I thought it would be nice to mention these on the screen. The university sponsor us. We have sponsors coming directly from iGem, for example. Yeah, I think it's nice to end it here and just go into the questions.
Are there any questions from the audience by any chance? Not, then if there are questions, please repeat them over the mic. But I actually have a question for you, because I didn't follow, I hope you didn't actually mention it already, because I had to walk out in a second. But I was wondering, because now you're going into the lab, like you're developing everything, for the competition, like you hope to have a valid product. Can you say anything about what happens after that? Say everything goes perfect, like you find like the solution that you have. Can you say something about how it how long it will take to implement, for example, in actual healthcare, or like how that process will go?
Well, we've already been looking into this also in the context of human practices, looking at the companies, for example, for developing the pill and stuff like that. That all has relatively realistic prospects, and there has always already been interest in seeing how that can be developed. However, the main setback we are currently looking at is the whole legislation around genetically modified organisms. The problem with that is just currently, especially in Europe, they are simply confined to the lab. So even if everything went perfectly and it had like a low limit of detection, high sensitivity, it worked perfectly, we're still limited or bound by the legislation of the GMOs in Europe, because they are simply kept in the lab. Even we want to work with E. coli, BL21, it's like a risk level 1, it's essentially the lowest risk level of GMO you can work with, yet still they have to be maintained in the lab. We do plan, and we have some, how do you say this, contact persons where we wanted to go into discussion with legislative individuals in Brussels right now. For example, with Europa Bio and Holland Bio, which are specifically companies that work on legislation on GMOs, for example. But you have to realize that, for example, clinical testing, etc., is in the context of tens of years very often. So hopefully, by the time the product is actually developed to the point where it works, legislation has also caught up with that. But that is currently just the major setback, because we are limited by the legislation. However, that doesn't stop us from doing the proof of concept, because I think this is really important, just showing that it has potential. Using cells essentially as living sensors, for example. Biology has given us the tools, highly optimized enzymes to detect things, for example, to auto-regulate itself, to produce proteins by simply giving food. Like nanomachines, for example, simply can't do that right now. Biology, in my opinion, has given us perfectly the tools to work with it now. CRISPR-Cas has allowed us to make whatever gene edits we want, for example. So it is, in my opinion, important that we show that it's possible, even if currently we can't translate it into a product. And I think also that's why we join in as a student team and we join in a competition, because I think that's currently in academia the most important thing we can do, not necessarily making a product.
Great, thanks. Also, another question. I see it's funny because we have the mobility easy mobility lab. We also have them coming up as a speaker. Can you say a little bit how AI is involved? Because I mean, that's an AI lab, right?
Well, it's a bit of a technicality because the AI Systems Institute is actually just our direct sponsor from the university. It used to be strategic areas, but I think they were dissolved. So we mentioned them as a sponsor, and we also coordinated by them, of course, and they help support us with networking with other teams, for example. But AI directly, for example, is not involved in our team, I'd say.
Okay, fair enough. Any more questions from the audience? Yeah? Do you want the mic or?
So to reiterate, like if we have connections with the FDA, if we are approved already, or? We haven't directly addressed medical companies like Pfizer, for example, yet for this product, particularly. We have been approaching companies to see, like I said, for the development of a pill, for example, to see what the prospects are there. But for example, FDA specifically, we haven't looked in yet, mostly because, you know, that's also something you contact when you actually want to have your treatment, your diagnosis approved by the FDA. That's for us just really, really far away. Also Pfizer, I don't think we have contacted Pfizer particularly. What we do want is, like I said, for human practice, we want to see if companies are interested for giving us feedback, for working together with us. But those specifically, no. Does that answer the question, or did I misinterpret?
No more questions from our audience? Nope. Okay, then Luc, I want to thank you for for joining us as a speaker, and we're just going to round it up. I'm just walking to the screen. Hi, I'm back. So, Luc, thanks for your presentation. For the people watching at home, thanks. Also for the people that are rewatching, thank you. For everyone at home and here, I want to also point out that next week our presentation is going to be done by Frans van der Vossen. He's going to talk a little bit about artificial wombs, just like growing babies outside of a human body. So that's going to also be a very interesting talk. For now, I want to invite everyone here to the drinks. We also have Innovation Café special dinner afterwards that you can join. Luc will also have some food with us, right, Luc? So you can join as well. It's a Innovation Café special. It's a meat, fish, or vegetarian dish. Meat and fish for 17.50 and a vegetarian dish for 15 euros if you want to join. And for now, I just want to invite you all to join us for the drinks and see you in the bar. So, thanks.